Okay, the specific defenses are activated by exposure to antigenic fragments, so they're not going to become activated until, for example, you have some cells that are for the chickenpox, and they're not going to become active until they notice chickenpox in your body. The T and B cells must encounter their antigen, and usually this is through antigen-presenting or dendritic cells before they are activated. And then once they're activated, they will destroy it. But this keeps them from being active when they're not needed. So this shows kind of generally how they're activated or how they're exposed to the antigenic fragments. So we have what are called major histocompatibility complex proteins, or MHC proteins for short. This shows the MHC1 proteins. MHC1 proteins are present in all cells. When a cell becomes infected by a virus or a bacteria, as we see is happening here, the MHC proteins will grab the foreign protein, so they're grabbing the foreign protein, and it's going to be sent to the plasma membrane through a transport vesicle. And now this is like putting up a flag on the cell saying, "Help, I'm infected, come get me." So they're going to alert the immune cells. So that's all cells have these MHC1 proteins. And then special cells have the MHC2 proteins. They're found in the antigen presenting cells, or APCs for short. All phagocytes and macrophages. So phagocytes and macrophages are considered antigen presenting cells, and they're going to have the MHC2 proteins. And these will actually go out looking for foreign things. The phagocyte will engulf the pathogen. As you see here, it will destroy it with a lysosome. And then the MHC2 proteins, they're going to go and take these antigenic fragments, these parts of the foreign cell, and take them out to the plasma membrane where other cells can recognize them. And they might take them to a lymph node or somewhere where our body has lots of T cells and B cells, and this is where those T and B cells would become activated. So T cells have receptors that recognize class 1 or 2 MHC proteins and a specific antigen. And that's what we see here. And that's how this cell is going to be activated. So antigen recognition. If the T cell recognizes the antigen, it will bind to the infected cell. We need to know about CD markers, which CD is cluster of differentiation. CD8 T cells recognize class 1 MHC proteins and their antigens. And then CD4 T cells, these are going to recognize class 2 MHC proteins and their antigens. Okay, a couple of review questions for you. Immunity that results from exposure to an antigen in the environment is classified as, and since you're exposed to it and it's in the environment, that's going to be active, naturally acquired immunity. Immunity that is present at birth is called, and that is called innate immunity. You're just born with it. You don't have to do anything to get it. Alright, next we're going to talk about antigen recognition. So a CD8 T cell recognizes a class 1 MHC protein and its antigen and binds to it. And then we have what is called co-stimulation. That's the next step. In co-stimulation, a T cell must be physically or chemically activated by the infected cell. And this step keeps the immune system from attacking our own cells. So this T cell is only going to become active when this particular pathogen is present. Once the T cell is activated, it's going to divide rapidly into, first of all, cytotoxic T cells. The cytotoxic T cells are the ones that seek out and destroy cells bearing their target antigen. These cytotoxic T cells can release perforins, which destroy the target cell's plasma membrane and cause lysis. They can also activate transcription of genes that cause apoptosis, which causes that cell to self-destruct. They can release lymphotoxin. Lymphotoxin will disrupt the metabolism of the target cell and kill it. So several ways for the cytotoxic T cells to destroy the target cell. Then the memory T cells, they're produced in case we're exposed to that same antigen again. We'll be able to mount a stronger and faster and better response to it and probably knock it out before we even know we're sick. And then we have the suppressor T cells. They provide a delayed suppression of the immune system so we don't attack our own cells. So once the pathogen is gone, it's going to turn off everything. Okay, so the CD4 cells are activated by exposure to Mhc proteins. The Mhc proteins, as we see here, bound to their specific antigen. So an example of that would be chickenpox. They will divide, the CD4 cells, they'll divide and produce helper T cells and memory T cells. B cells must also encounter their antigen to be activated, and this is called B cell sensitization. They're sensitized by binding their antigen and then displaying it on Mhc protein. So now it's sensitized. Then they bind to helper T cells and are activated by cytokines. So again, the helper T cells are needed to turn on the B cells and get them ready to go. Then once the B cells are activated, they'll divide to produce memory cells and active B cells, which are stimulated by cytokines to become the plasma cells. Plasma cells are the cells that actually produce the antibodies, and they can produce 100 million antibodies per hour, which is quite amazing. So let's talk next about antibodies. Antibodies are small soluble proteins that bind to specific antigens. They can inactivate antigens or they can activate other defensive processes. They're made up of 4 different peptides. They contain a constant and a variable region. So here's the constant region, and the variable region would be out here on the ends. The variable region contains the antigen binding site. So that's what's going to bind to the particular antigen, such as chickenpox. And the constant region is going to be the same for each class of antibodies, but we're going to have one antigen binding site for chickenpox, another one for measles, another one for the flu, whatever. But the constant part is important. The constant region has binding sites for complement proteins, macrophages, basophils, or mast cells, because we'll see that the antibodies don't actually kill cells, they just kind of tag them and mark them for destruction. So antibodies bind to what are called antigenic determinant sites on the antigen. Complete antigens will have 2 of these antigenic determinant sites like this. Partial antigens, or haptens, only have 1. So partial antigens do not normally bind to antibodies, but they can if there's some sort of carrier molecule. And if that carrier molecule is normally present in tissues, antibodies may begin attacking normal cells, and that can lead to allergies and autoimmune diseases. This shows the different classes of antibodies. Most of them are IgG antibodies, about 80%, so make sure you know that. In a primary immune response, this shows the antibody levels. The red is the IgM and the red is the IgG, and you can see it takes about 2 weeks to get our peak levels of the IgG antibodies, which are the ones that are actually going to be attacking the pathogens. And if we look at a secondary response, the response is much faster. It's also much stronger, and it's also longer lasting. So if we look at those together, we can compare. You can see to get the same level of antibodies takes only maybe less than a week in the secondary response. And that's because we have these memory cells. The memory cells can live 20 years or more, and so if they're already ready to go, they can start making antibodies right away, and that's why we can kill things right away. That's why we usually only get sick with something once, usually. Now, antibodies do not directly destroy antigens, but they use several mechanisms to eliminate them. One is precipitation and agglutination. So they'll just make things clump together, and if the antigens are bound together, they can't get into our cells. And it also makes them easy targets for phagocytes. In that video we saw the phagocyte having to chase things. If the bacteria are all clumped together, they can't move very well. Another thing they do is neutralization. If a pathogen is coated with antibodies, it cannot enter our cells, so it can't hurt our cells. So that's another thing they can do. Another mechanism they use is called prevention of pathogen adhesion. Antibodies in our saliva, our tears, our mucus, and our perspiration, they will prevent pathogens from attaching to body surfaces, and if they can't attach to us, they can't get into us. Another thing they do is activation of complement. So when the antibodies bind to their target, they change shape and they expose complement binding sites, and this will start the complement cascade that we talked about earlier, and that will lead to cell lysis and death. The next mechanism is opsonization. Phagocytes can bind more easily to antibody-coated pathogens, increasing their effectiveness. So I like to think of that as making the pathogens more tasty to the phagocytes. And then also just direct attraction of phagocytes. Antigens covered with antibodies will attract all types of phagocytes. And finally, stimulation of inflammation. They will stimulate the release of inflammatory chemicals from basophils and mast cells, and that will help with healing. So next we're going to talk about allergies. Allergies are inappropriate or excessive immune responses to antigens or allergens. So the first time you're exposed to an allergen, it will activate T and B cells and these IgE antibodies. These IgE antibodies will be produced. Then they sensitize mast cells and basophils. And if we're exposed to that antigen again and it binds to one of these sensitized mast cells and basophils, they're going to release lots of histamine, and that causes inflammation and itching. Now, at the body surface, this is just annoying, and it's what leads to like a runny nose, watery eyes. But if the allergen enters the bloodstream, this can lead to anaphylaxis. And the peripheral blood vessel dilation leads to a dangerous drop in blood pressure that can lead to circulatory collapse or anaphylactic shock. And this can also lead to death due to airway constriction. So we talked about the specific and the non-specific defenses separately, but they really do work together to defeat the pathogens. Macrophages that are antigen presenting cells, so they're part of the non-specific system, but they are required to activate the specific defense system. And antibodies from the specific defense system enhance phagocytosis and the complement system. Phagocytosis and antigen-presenting cells are the most effective defense against bacterial infections. With viral infections, cytotoxic T cells and natural killer cells can be activated by infected cells. So we need different mechanisms for different types of pathogens. Sometimes it's hard to recognize viruses because they get inside of our cells. So our phagocytes don't recognize them as readily. And finally, we're going to talk about autoimmune disorders. So the immune system normally recognizes and ignores self-antigens. But when the recognition system malfunctions, activated B cells make antibodies against normal body cells called autoantibodies. Thyroiditis is caused by autoantibodies against thyroglobulin, and that will mess up your thyroid gland. This picture is showing someone with rheumatoid arthritis. It occurs when autoantibodies form immune complexes in connective tissues around the joints. This leads to inflammation and eventually joint destruction. Insulin-dependent diabetes or type 1 diabetes is caused when autoantibodies attack insulin-producing cells in the pancreatic islets. Okay, this is not really an immune disorder, but more like an immune problem. So one problem with organ transplantation is we can have what's called graft rejection. Basically, your immune system, even though you need a kidney, I think that's a kidney, your body needs a kidney, but your immune system will recognize this transplanted kidney as foreign and destroy it. So if you get an organ transplant, they give you immunosuppressive drugs, and that will reduce the sensitivity of the immune system. Allergies are also autoimmune disorders, usually pretty harmless. Immunodeficiency diseases result from problems with the development of lymph organs and tissues, or infection with a virus that depresses immune function, or treatment with or exposure to immunosuppressive agents like radiation. So sometimes it's a developmental problem, sometimes it's a virus, sometimes it's things like radiation or chemotherapy. AIDS is the most common immunodeficiency disease. It's caused by the HIV virus, which infects helper T cells. And this is a problem because we need those helper T cells to activate the rest of the immune system. So if our helper T cells don't work, nothing works. So the gradual destruction of helper T cells impairs both cell-mediated and antibody-mediated immunity. And with our immune function suppressed, ordinarily harmless microorganisms can invade and cause what are called opportunistic infections that wouldn't hurt a normal person, but in this person with their immune system compromised by the disease, that can be lethal. So you don't actually die of AIDS, you die of some infection that your body's not able to fight off because of the AIDS. And as we age, the immune system becomes less effective. There's a reduction in the level of thymic hormones, and that leads to a reduction in the number of helper T cells. Immunological surveillance also declines, leading to increased rates of cancer.